when you look at human chromosomes under a microscope, you see that there are 46, which is 23 pairs, and they're all very different from each other, so they're identified by their size, their shape, and their staining pattern. A karyotype is the entire set of chromosomes arranged in decreasing order. So number one, your two number ones are the largest, and then getting smaller and smaller and smaller, all the way down to pair number 22, which are the smallest. Now, these are what are called autosomes, because these are the chromosomes that contain the genes for making the human and doing everything that the human needs to do. So autosome, auto meaning self or of the body. That 23rd pair is not arranged by size and shape because they're very different. These are your sex chromosomes. So in the human, of course, the sex chromosomes are that 23rd pair, and females have 2 copies of the X, and males have a copy of one X and one Y. So that Y chromosome is the smallest of all chromosomes and contains just over 200 genes. So if we look at how it's actually the sperm cells that determines the sex of the body, we can put that onto a Punnett square and prove it. So remember, we're looking at just the 2 chromosomes that are the sex chromosomes. So all females have 2 copies of the X. So when the female is undergoing meiosis to create those oocytes, every cell is going to get a copy of the X chromosome because that's all mom has, right? That's all the female has. The males have one X and one Y. So in the process of making sperm cells, 50% are going to get an X and 50% are going to get a Y. So all female eggs contain one X chromosome. 50% of male sperm have an X, 50% of male sperm have a Y. Okay? So if you do your Punnett square and you carry all of your X's down and all of your X's across and your Y's across, you will see that there is indeed a 50% chance of this X chromosome fertilizing an egg, and we know an egg's got to have an X, and there's a 50% chance of this Y fertilizing an egg, and we know the egg has to have the X chromosome. So this shows you that it is that Y carrying sperm cell that determines the sex of the baby. If it is to be male, the Y sperm cell fertilizes the egg, and if it is a female, the X chromosome fertilizes the egg. So regardless whether fertilization is by an X chromosome or a Y chromosome, both male and female embryos develop identically until about 7 weeks after fertilization, and it's at that point that one or more genes set into motion a cascade of events that's going to lead to the development of a male. If that doesn't happen, if the genes are mutated or those genes are absent, then the female pattern of development is going to happen. It's been known since 1959 that the Y chromosome is needed to begin the male development embryonically. But experiments published in 1991 showed that there is an area on the Y chromosome called the SRY, and that's the sex-determining region of the Y chromosome. And it has genes that are commonly called the TDF genes, testes determining factor. So when a small amount of DNA fragment containing this gene was inserted to 11 female mouse embryos, 3 of them developed as males. Now, the other ones just didn't develop at all, okay? So the SRY acts as a molecule to switch or turn on that TDF gene, and that TDF gene causes the testes to develop. So only if the SRY gene is present and functional in a fertilized ovum will the fetus develop testes and differentiate into a male. Without that functional SRY, the fetus is going to develop ovaries and differentiate into a female. So there's some case studies that have confirmed the role of the SRY in directing the pattern of male development in humans. In some cases, phenotypic females with an XY genotype were found to have mutated SRY genes. So they were actually genotypically male because they had an X and a Y. But because the SRY gene wasn't working, these individuals failed to develop normally as males. In other cases, phenotypic males with an XX genotype, remember XX genotype is female, but they developed as a male, so they were found to have a small piece of the Y chromosome that included the SRY gene had gotten inserted into their X chromosome, probably during the process of crossover. In addition to determining the sex of the offspring, the sex chromosomes are responsible for the transmission of several non-sexual traits. Many of the genes for these traits are present on the X chromosome, but are absent on the Y. So these features produce patterns of heredity called sex-linked inheritance. And that's a little bit different than the patterns of inheritance that we've already talked about. So color blindness is one example of these sex-linked disorders. It's actually the red-green color blindness is the most common type of color blindness. So this condition is characterized by a deficiency in either the red or green cones that are responsible for color vision in the eyes. So red and green are seen as the same color. But the gene for red-green color blindness is a recessive one, and it's designated as lowercase c. Okay, so here you can see lowercase c. Okay, there's our normal gene. There's our lowercase c, so this is a carrier with one dominant gene. And then we don't have anything on the Y because that gene is not on the Y. So for normal vision, designated as dominant C, uppercase C, you're going to have normal vision. But the uppercase C, lowercase c, remember that's heterozygous, those genes are all going to be, the dominant and the recessive copy of these genes, whether it's the dominant copy or the recessive copy, are only located on the X chromosome. So the ability to see colors is only on the X chromosome. So only females who have 2X with the recessive gene are going to be colorblind. But this is a very rare condition, can only result from the mating of a colorblind male and a colorblind female or a female carrier, because males don't have that second X chromosome that could mask the trait. All males that are born with an X recessive gene are going to be colorblind. There are also some sex-linked traits, and the most common of these is hemophilia, which we commonly call free bleeding. So this causes the blood to clot very slowly after an injury. So it's a sex-linked trait. And like the trait for colorblindness, hemophilia is also caused by a recessive gene. Other sex-linked traits, let's try that again. Other sex-linked traits in humans are Fragile X syndrome, a condition that causes sweat glands to not be functional, certain types of diabetes, certain types of deafness, the absence of some of the incisors, night blindness, some types of cataracts and glaucoma, and also juvenile muscular dystrophy. All right, so we want to start looking now at a little bit of disorders associated with the reproductive system, and one that is very common is infertility. So in female infertility, that means the inability to conceive, of course, occurs in about 10% of all women that are reproductively capable in the United States or of reproductive age. Female infertility can be caused by ovarian disease or obstruction of the fallopian tubes or anything that prevents the oocyte from being fertilized. But it can also be when the uterus is not being prepared adequately to receive that fertilized ovum and for implantation to occur. Male infertility is typically called sterility, and that's an inability to fertilize an egg. It does not mean that erectile dysfunction or impotence is going to occur. So male fertility, remember, requires that there's a whole lot of viable sperm cells that are going to be produced by the testes, but then they also have to be able to get through all of the duct system and be deposited into the vaginal canal if fertilization is going to happen. So the seminiferous tubules of the testes are very sensitive to a lot of factors such as x-rays and infections, toxins, malnutrition, and even higher than normal scrotal temperature. So all of this can cause a decrease in the number of sperm count or degenerative changes in the way that the sperm are made. One cause of infertility in females is inadequate body fat. So this develops a lot in extreme athletes or females that have developed anorexia or bulimia, and their fat content decreases below 10 to 15%, what is normal for their height. So this can delay the onset of menstruation. It can inhibit ovulation during the reproductive cycle. It can cause amenorrhea, which simply means that the periods stop. So both dieting and intense exercise can reduce body fat below the minimum amount that is required for sufficient levels of estrogen in the body. So how do you treat this? Well, the most common way is simply weight gain or reduction of exercise so that a little bit of fat does again accumulate in the female body. But not just the women with too little fat. There's been studies that also show that very obese women experience problems with amenorrhea and infertility as well. Males also experience reproductive problems in response to malnutrition or undernutrition and weight loss as well. So, for example, they produce less prostatic fluid and reduced numbers of sperm, and the ones that they do produce may also have reduced motility. But there are many techniques now that can be used to assist infertile couples in getting pregnant and having a baby. And the first time this was recorded was back in 1978 in England, where there was the first case of in vitro fertilization. So the mother was given a lot of follicle stimulating hormone, and then of course she developed a lot of secondary oocytes rather than just a single one like would be normal. And so this produced something called superovulation. So when several of those follicles had reached a large enough size, there was an incision made, the follicles were removed, and they were transferred into a solution containing sperm, and the oocytes underwent fertilization. So hence the term test tube babies. When a zygote is achieved by in vitro fertilization and it reaches about the 8 or 16 cell stage, then it's implanted into the uterus, and if all goes well, then development continues as normal. Now, of course, mom's going to have high levels of estrogen and progesterone so that uterus is going to become thick and gooey, very vascular for implantation to occur. Another way another treatment for infertility where that infertile couples can have a child, or I should say another option for infertile couples, is called embryo transfer. So a man's semen is used to artificially inseminate an oocyte from a donor. Then after fertilization, in the donor's uterine tube, the morula or the blastocyst is going to be transferred from the donor into the infertile woman, who can then carry it to term. Embryo transfer is usually done for women who are infertile or have a genetic defect that they have found that they're a carrier for, or some type of serious genetic disorder, and they don't want to pass that on to their offspring. All right, so congenital simply means born with. So there are abnormalities that are present at birth, and usually before birth as well, and these are called congenital defects. So some of these defects occur during the formation of structures that are developing during the period of organogenesis. And remember, that's about the 4th through the 8th week of development when all the major organs are forming. During organogenesis, the stem cells are establishing the basic patterns of organ development, so it makes sense that it's during this time that the developing structures are going to be very susceptible to genetic and environmental influences. Major structural defects occur in about 2 to 3% of live-born infants, but they're the leading cause of infant mortality and accounts for about 21 to 22% of infant deaths. Many of these defects can be prevented by supplements or avoiding certain substances. For example, neural tube defects, which cause the spinal cord and the vertebral column not to develop well. For instance, spinal bifida or anencephaly can be prevented by having a pregnant woman take folic acid. Iodine supplements can prevent mental retardation and bone deformation that's associated with cretinism, or congenital hypothyroidism is the better term. And of course, it should make sense that avoidance of any teratogens is very important in preventing those congenital defects. Down syndrome is a disorder that's characterized by 3 copies of chromosome 21, so it is also called trisomy 21. About 1 infant in every 900 to 1,000 is born with Down syndrome, but older women are more likely to have a baby with Down syndrome. The chance of having a baby with Down syndrome is less than 1 in 3,000 for women under the age of 30, but it increases to about 1 in 300 between 35 and 40, and then after the age of 48, it increases to about 1 in 9 children. So Down syndrome is characterized by mental retardation and retarded or depressed physical growth, such as short stature and stubby fingers, very distinctive facial structures, such as a large tongue, a flat profile, very often slanted eyes, and a rounded head with a broad skull. Usually accompanied by kidney defects and depressed immune system, malformation of the heart, the hands, the ears, the feet. Very, very rarely do these individuals attain sexual maturity and are able to reproduce, and their life expectancy is also typically much shorter. And then finally, some miscellaneous terminology associated with the reproductive system. So fertilization age is 2 weeks less than gestational age, because a secondary oocyte is not fertilized until about 2 weeks after the last menstrual period. So gestational age is the age of an embryo or fetus as calculated from the last day of the menstrual period. And of course we know that ovulation happened about 2 weeks after the last menstrual period. So the difference in those two terms. Just a few others that I want to mention while we're talking about especially the sex chromosomes. There is a condition called the triple X syndrome or meta-female, where the sex chromosome is characterized by having at least 3 X chromosomes, and that occurs about 1 in every 700 or so births. These females usually have underdeveloped genitalia and limited infertility, and may also be mentally retarded. In some cases, though, the female appears very much normal, but does have more of the developmental, the mental developmental characteristics. And they may be taller than average, disabilities with speech and language, weak muscle tone, and often behavioral and emotional difficulties. Klinefelter syndrome is a sexual chromosome disorder that's due to trisomy, but it's 2 copies of X, so XXY. Now that occurs in about 1 of every 500 births. The people that have this usually are somewhat mentally disadvantaged, and males will have underdeveloped testes, less body hair, and usually also a little bit of enlargement in the breast, poor growth of facial hair, testicular atrophy or decrease in size. And then finally, there is Turner syndrome. So Turner syndrome is a sex chromosome disorder caused by the presence of only 1 X chromosome. So we designate that XO or X0. This occurs in about 1 in every 5,000 births, and it produces a sterile female with virtually no ovaries and very limited development of secondary sexual characteristics. So typically you see a very short female with somewhat of a thick webbed appearance to her neck, underdeveloped breast. The nipples may be widely spaced, but intelligence is usually normal. All right, I think this concludes our lecture on reproductive system. We'll talk again soon.